Hello, welcome. Come on in, everyone. Welcome. We're just a minute or two before 4 o'clock, so. I hope everyone's doing well today. Got a little bit of snow, just a little though, nothing too bad, which is nice. If you would, go ahead and type your first name into the chat for me so I can take attendance. Thank you. Thank you very much. That's great. Thank you. Okay. 3, 4, 5. Still missing a couple more. We'll give them another minute or so. Today in lab, we're going to be talking about our next subject, which is simple staining. We'll pick up from where we left off discussing basic wet mounts and the hanging drop technique. And we'll talk about how we do what are referred to as simple staining procedures and why we do them. And we'll take a look at some slides with microbes on them that have been put through a simple staining procedure. And we'll practice looking at different shapes of bacterial cells particularly. I think I mentioned to you that I have a couple of dogs. This is Miss Millie. Say hi, Millie. Say hi, look at the camera. This is Millie, she's 2. She's generally pretty good. But, you know, she thinks she's the neighborhood police officer. So anytime she hears any kind of commotion outside, she goes a little crazy, right? You're a good
Excuse
dog.
me.
Yeah.
Can I ask a question?
Sure.
So if we did get the book, is there any way you could tell us what page we would be on?
So if you pull open the syllabus,
okay.
At the end of the syllabus, there's what we call the tentative schedule. Let's see if I can pull it up for us here. And that shows week by week what we're going to be doing. Hold on, I'll pull it up for us so we can look at it together.
Are you talking about where it has like the dates and that
yeah,
stuff?
Exactly. So right here, I'm pulling up, this is the last page or the second to last page of the syllabus. So the information about the course is in the early parts. And then if you go to the very back, that's where the tentative schedule is. So it's organized by Mondays and Wednesdays, but remember, we're working generally by week in this course. And you can see that in addition to the video topic, there's also the text chapter that's associated with that topic. So this week, well, the first week, all the introduction material you could read up some more about in chapter 1. And then this week, we also go into chapter 2 and chapter 3 in the text. And remember, you're going to want to use that text sort of as a supplement. So as you read through it, if there are things in the text that, we haven't discussed, they're interesting to read about, but don't worry about, taking copious notes or anything, because you're not going to be tested on anything that we don't talk about in class, in the lecture videos. Okay?
Perfect. Thank you.
It's a very good textbook, but it is a very, it's It's a very thorough textbook, and there is a good amount of material in that text that we just don't have the opportunity to cover in a course like this one. All right, very good. All right, if you haven't yet, if you could type your name into the chat, that would be great. So I have a record, I can get a count. I think we're still missing a couple of people. All right. Oh, very good. Okay. All right. So let's start off today by asking if anybody has any questions about any of the lecture material that you may have watched so far, both last week's material and also what you're watching this week. You may not have watched it all yet, but the history of microbiology material and the science of microscopy, and also the lecture that's called The Cell, which talks about some basic cellular structure. If anybody has any questions, feel free to type those in the chat and we can talk about those. Just generally speaking, each week, what I'll do is on Monday, at the start of our lab on Monday, I'll go over any of the quiz questions from the previous week that people may have missed. We won't talk about the quiz, like for example, today, we won't talk about either of this week's quizzes because there are people who still haven't taken them. So in fact, most people haven't taken them because it's not time yet, but that's what we'll plan to do on Mondays first thing. So what we're going to be doing today is an exercise about a process known as simple staining. And I'm going to go ahead and share that with you. Remember that I do post just the slides up on Canvas for you. If you find that helpful, you may or may not. I know some students like to pull slides up and actually use them to take notes on. So those are available if you need them. But again, remember that any slides that I post on Canvas for you are not going to have any of the video clips that we look at during lab. So if you need to go back and look at video clips or re-listen to anything that we may have discussed, you're going to want to go to our playlist on YouTube and pull up the lab video, and then you can review anything you need to there. So what we're going to be doing this afternoon is we're going to first just talk about staining in general, a little bit of the science behind staining, and there are some vocabulary terms that we need to learn today. And then in the second part, I'm going to show you how to prepare what's called a smear, how to heat fix it, and how to do a very simple, what's called direct staining procedure. And then we'll spend a few minutes towards the end of class today looking at some slides with bacteria on them that have been put through a simple staining procedure. And we can practice identifying the shapes of bacteria. So let's spend a couple minutes first talking about stains and introducing some vocabulary about stains. We use a lot of stains in the microbiology lab. The only other lab that uses so many staining chemicals is the pathology lab. We have so many stains because each stain is designed for different types of microbes. Each stain is designed to highlight different structures in a cell. Some stains are designed for very particular types of microbes, and some you can stain virtually any microbe with. In order to prepare a sample of a microbe on a glass slide, on a microscope slide, the very first thing we have to do is create what's called a smear. And when I use that term, with regard to microbiology, a smear is simply a thin layer of cells that I've placed onto a microscope slide. And we're going to look at a video clip to see how we go about spreading those cells out on a glass slide. After you make a smear in the laboratory, you have to go through a process called fixation, before you stain those cells. Now, the fixation process does two things. Most importantly, it's going to make those cells stick to the slide. It's going to cause those cells to adhere to the glass on the slide. And the reason that's so important is because when we go through the staining procedure, we're going to be rinsing liquids on and off of that microscope slide. If you forget and you don't fix the cells to the slide, you will simply wash them away. And believe me, I've done it. Plenty of other students have done it. You have to remember the fixation step so that you don't accidentally flush your precious cells down the drain. The other thing to remember, and I mentioned this last time when we were talking about biosafety, fixation is also going to kill the cells. Both the fixation process and the staining itself is going to kill whatever cells we put on a microscope slide. So that's important to remember. Now, when we go to fix a smear, there are two ways we can do it, and both ways are equally valuable in the laboratory. Different people have a different preference as to which one they like, but both of them are equally easy to do and do just as good a job. The first thing we can do is called chemical fixation. We use methanol for that, and we specifically use a mixture of 95% methanol. Now, it has to be methanol. And while there are lots and lots of alcohols that we use in the laboratory, methanol is one that's very toxic. So you have to be careful when you handle methanol. Of course, you want to have your gloves on as usual. But a 95% solution of methanol can just be placed onto the slide with a dropper and let to sit for a minute or two. And that is going to fix the cells to the slide. Now, the other method that we use for fixation is called heat fixation. And like the name suggests, we just place the slide into some sort of a heat source for a very short period of time. And what the heat will do is literally warm up the membrane around the cells so that it helps it stick to the glass slide. Now, you need to understand what's happening when we fix a cell, because we are changing a cell just a little bit when we heat fix it or chemically fix it. What both of those things are doing is coagulating some of the protein that's present in the membrane around cells. And by coagulating it, it makes it a little bit sticky. Now, there's lots of proteins, lots of different proteins that are sitting in the membrane of a living cell, including your cells and my cells. But particularly, there are enzymes that end up getting coagulated during this fixation process. And what happens with the enzymes is important, because by coagulating, or to use a more scientific word, by denaturing enzymes in these microbial cells during fixation, it's going to help us prevent a process that could go on, which would destroy the cell. Now, let me say that again, because I know that's a little confusing. When you fix a slide, when you fix the cells to the slide, you attach them to the slide with either chemicals or heat. What you're really doing is you're coagulating protein that's in the membrane, and you're making it sticky, so it literally sticks to the slide. But the other thing you're doing is you are denaturing or destroying a specific kind of protein called enzymes. Remember, enzymes are what do all of the metabolic reactions inside of a cell. When you denature enzymes, what you do is you prevent them from doing their normal work. just like if you took a piece of, let's say, chicken breast and you cooked it, you're denaturing it, you're changing it. And when you denature these enzymes inside of microbial cells, you basically inactivate them. And that's a good thing, because if you didn't do that, what would happen is those enzymes, as that cell was getting stained, those enzymes would start to degrade the structures inside the cell. And you would end up with a damaged, very odd looking cell when you finally put your slide underneath the microscope. So fixation is important. It not only helps keep cells on slides, but it also helps maintain the structural integrity of those cells. So we'll get a good examination of them. Now, when you're doing the chemical fixation, like I said, you're simply placing a drop of methanol on the slide over the place where you put the cells. When you're doing the heat fixation, it's a little bit different. You have two basic methods, at least two that most people use. Number one, you can simply pass the slide through an open flame. What you do is you take a basic a clip, any kind of a clip, but most people use wooden clothespins in the laboratory because they're very helpful. You put your slide into the clothespin so that you don't have to hold the slide close to the flame. And you simply pass your slide through the flame 3 or 4 times. So you're not holding the slide in the flame. That would actually be bad. You'd actually burn up all your cells. You're just passing it across the flame to get the heat. Now, there's also a device that's used in microbiology laboratories called an incinerator, and I'll show you what this looks like. This device is useful because it means we don't have to have an open flame in the laboratory. So it's a little safer than using a Bunsen burner or something. The incinerator itself has a metal casing, and the metal gets hot when the incinerator is heated up. So all you have to do with your slide in your clothespin is place the slide against the outer housing of the incinerator just for 20 to 30 seconds generally. And that will be enough to heat fix the cells. Now, before I show you what this looks like, I have a note at the bottom of the slide that's really important. Put a star next to this. It's really important that when we make a smear, we wait for the smear to dry before we do our fixation process. Now, this doesn't really matter with chemical fixation, but if you're going to use heat to fix your slides, you've got to wait for the smear to dry. And that's because even if it's a little bit damp and you put that slide over heat and you heat up the cells, you can create an aerosol. You won't see it. It'll be very, small. The amount of material that you're sending up into the air will be very small, but it's enough to be dangerous. So we only fix, specifically heat fix, dry smears. All right, very important safety note there. All right, on this slide, down at the bottom, you can see a Bunsen burner with a nice blue flame. You can see over to the right, the silver looking device. That's just a pair of hemostats that someone is using to hold their glass slide that has a smear on it. You can see the smear. You can't always see a smear on a glass slide, but here you can. Now, this is kind of a silly image because the person is holding the slide over the flame and it's just staying there. And we would never do that because within about 5 seconds, all of the cells would just be gone. You would have burned them to a crisp. Instead, what you do is you pass the slide back and forth through the flame 3 or 4 times. And that's plenty of heat to fix your cells. Remember, always wait for the smear to dry before you apply heat to it. If you look up at the top, there's a device here, this black sort of rectangular device. This is called a slide warmer. And any modern hospital-based laboratory, microbiology laboratory, is going to have one of these. You'll notice that there's a bunch of smears sitting on this, basically a hot plate. What they do is they set this plate to a very low but warm temperature, and they put the smears on the plate to help them dry faster. It's not going to fix these slides, but it's going to dry them enough so that they can then use a flame to fix them. All right, I want to show you, first of all, just a glass slide that's attached to a clothespin. Very low tech equipment here. You'll see the clothespin is colored with pink and purple and blue because it's been through a lot of staining procedures. But the reason we use something like this, a clothespin or a hemostat or any other clip, is because we want to keep our fingers away from flames, obviously. So this allows us to do things with slides without getting our fingers too close to them. And the other thing I want to show you is what an incinerator looks like. This is a basic incinerator. So we plug this in and turn it on, and it has a cavity through the center. It has a tunnel through the center that's made out of ceramic. And that ceramic tunnel gets very hot. It gets red hot, in fact. And we use that tunnel while we're working in the laboratory to heat sterilize our metal tools. So if I take my loop, for example, my metal loop, and I put it into a culture, I can then put that loop into the incinerator. And within a matter of a few seconds, I can sterilize the loop so I can use it again. Now, the outer housing on this incinerator is made out of metal as well. And so it's going to also heat up. It's important to know to be careful with incinerators when they're on your desktop. While they're plugged in and heated up, you want to be careful you don't accidentally bump your arm into them because you can burn yourself on one of these. But they're also helpful when we need to heat fix. If you don't have a Bunsen burner or you don't want to use a Bunsen burner, you can simply take your glass slide and just lay it against the metal for 20 to 30 seconds, and it will be hot enough to heat fix your cells for you. So you'll see the incinerator again as we go through the semester. They're more and more popular now in laboratory spaces because they're safer. They're safer than open flames. Anybody have any questions so far? Any questions about anything I've talked about so far? Okay, I'm going to go back now and show you the next slide. Let's take a minute now and talk just a little bit of vocabulary about stains. Most of the stains that we use in a microbiology lab are made out of salts. Now, this word salt makes everybody think about table salt or sodium chloride. But in chemistry, all a salt is a combination of two ions. So sodium is an ion, it has a positive charge, and chloride is an ion, it has a negative charge. When those two things come together, you get table salt, which is sodium chloride. And of course, the two charges cancel each other out. So any salt that you can name is made out of two ions, a positive ion and a negative ion. So you could have things like calcium chloride or magnesium chloride. There's lots and lots of different salts. Now, these stains that are made of different kinds of salts, they also have ions within them that are colorized. We give them a particular name. We call those ions chromophores. And I have the word written out for you here. Anytime you see that term chromo, think color. So a chromophore is a molecule that has color in it, natural color. And any salt that we can use as a stain is going to have one of those, it's going to have an ion in it that has this color, natural color to it. Now, some chromophores are positively charged and some chromophores are negatively charged. Remember, they're ions, and all an ion is a charged atom. So whether it's an atom of sodium or an atom of chloride or an atom of calcium, if it has a charge on it, positive or negative, we call it an ion. So if a stain has a positively charged chromophore in it, we call that a basic stain. If it has a negatively charged chromophore in it, we call that an acidic stain. Now remember, those terms, basic and acidic, relate to pH. And just as a quick review, remember the scale for pH runs from 1 up to 10. The lower numbers are the acidic values. The higher numbers are the basic values. 7, a pH of 7 is considered neutral. So anything below 7 is an acid, and anything above 7 is a base. So some of our stains have positive chromophores in them, positive colorized ions in them. Those are basic stains. Some of our stains have negatively charged colorized ions in them. Those are acidic stains. And different stain types, basic or acidic, are good for different purposes. It turns out that most of the stains that we use for bacteria are basic stains. Most of the stains that we use to stain bacterial cells, which is what we look at the most, those are basic stains. So those have a positive chromophore in them. And of course, a positively charged thing is going to be attracted to a negatively charged thing. And inside of a bacterial cell, there are lots of things that are negatively charged, including things like the DNA molecule. So if you put a basic stain onto a bacterial cell, it's going to bind to anything that has a negative charge. There are negative charges in the membrane, and there are negative charges, like I said, in the DNA, and you're going to get that stain to stick because it'll stick to negatively charged things. Now, today we're going to be talking about simple staining, but there's another kind of staining we'll talk about in another lab called differential staining. When you hear somebody talk about simple staining, all that means is that the process they're going to use uses only one stain. In other words, we're only going to apply one color, one dye, essentially, to the cells. If we're using a differential staining procedure, we're going to apply several different stains to the cells, and each stain has a different color. Now, in the simple staining group, those are the procedures, remember, that use one stain. We can do what's called a direct stain, and we can do a negative stain. Some more vocabulary for us. A direct stain is a simple stain that stains cells. A negative stain is a simple stain that stains the background of the slide and not the cells. Direct staining is much more common than negative staining. Now, differential staining, like I said, uses different stains, a couple of different stains. They're each a different color. We're going to talk about a very common differential staining procedure next week or the week after, I can't remember, called gram staining. But today we're just going to stick with simple staining. One of the most commonly used simple stains that produces a slide with stained cells, so it's a direct stain, is one called methylene blue. Methylene blue is a basic stain. It has a positive chromophore in it. And the chromophore has a very distinct blue color. So it's a basic stain because it has a positive chromophore in it, a positive colorized ion in it. And it's a direct stain because it stains the cells, not the background. Methylene blue is very commonly used. It's very inexpensive. You can find it in virtually any laboratory. We not only use it to stain bacterial cells, but we also use it to stain other types of cells. So you would find it in pathology labs and things like that as well. Questions so far? All right, very good. So just some vocabulary for us around stains. We use them so commonly. It's important that we can communicate about them with each other and know these terms. All right, what we're going to do now is watch a video that shows us how to prepare a simple stained slide. It's about 15 minutes long, I believe. We start any of our work in the laboratory. Remember that we have to disinfect the work surface. The two most common disinfectants used in laboratories for this. Oops, let me try again. Sorry. We start any of our work in the laboratory. Remember that we have to disinfect the work surface. The two most common disinfectants used in laboratories for this kind of disinfection are alcohol and bleach. We use an alcohol solution of anywhere from 60 to 90%. Typically, it's about 70%. And we use a 10% bleach solution. Both of those things are rapidly bactericidal. They will kill bacteria on surfaces very quickly, and that's why they're so effective. So with the squirt bottle like this, we simply apply the disinfectant to the surface and then clean it off. Make sure the surface is nice and dry before you get yourself going with your work. Remember too that you have to make sure you have all of your materials gathered before you begin. And first and foremost, we want to glove up. These are nitrile gloves that I'm wearing in this picture. I put this picture in because I want you to see that these gloves are a little too big for my hands. You want to wear gloves in the laboratory that are not too tight or too loose. And generally speaking, when you look at the back of your hand, which is what you're looking at in this picture, you should be able to move your fingers and your thumb around and not get these very large folds. You don't want the glove to have a lot of extra space. You want it to fit against your skin, but you don't want to wear a tight glove. It's not good. It will cut off your circulation. On the palm side of my hand, you can see there's a lot of extra room in this glove. You can see the folds on my palm and along my fingers. But unfortunately, beggars can't be choosers, and right now this is the only size glove that I have available. Okay, so I've gathered the supplies that we're going to need today in order to make a smear of bacteria for staining. And what you're seeing on the desktop here, I've got a bowl that is used only for staining purposes. I've got a dropper bottle with methylene blue in it, which is a very commonly used simple stain. I've got a clothespin here, and I'll show you what we do with those while we're staining. I've also got my inoculation loop. This is an instrument that we use very often in the microbiology lab. It has an aluminum handle, and then it has a wire. And at the end of the wire, you can't really see it here, but at the end of the wire, there's a loop. When you dip this wire into a culture, into a broth culture, you will pull up some of that culture within the loop. If you remember when you were a little kid and you used to play with soap bubbles, you would stick that little wand down into the soap bubble solution and you would pick up some of the solution inside the loop. And that's what we're doing with an inoculating loop as well. Over on the right-hand side over here, you can see an incinerator. This is an electric device that gets hot enough to allow us to sterilize the wire on an inoculating loop. Now, in this test tube, I'm holding a nutrient broth culture. It has bacteria growing in it, so I would have just pulled this out of the incubator, and I'm now ready to examine cells that are inside the culture. Anytime you pull a broth culture out of the incubator, one of the indications that you have been successful in growing bacteria is the cloudiness that you see in the broth solution. The broth starts off very clear. You would be able to hold a piece of paper behind it and read the writing on the paper. But obviously this solution is no longer clear because it has cells growing in it. Now, it's really important that when you're using an incinerator in a microbiology lab, that you go ahead right at the start of your day and plug that thing in. These do take a few minutes to warm up, and they have to be red hot in order to work correctly. So I always plug the incinerator in right away when I get into the lab and I'm ready to start making slides. Note too that a lot of incinerators have two settings. They've got a low heat setting and a high heat setting. Now, I happen to be using the high heat setting today, but depending on the incinerator, the low heat setting might be just fine. Incinerators can get very hot, and sometimes having the incinerator on the desktop on high is just unbearable because it's radiating so much heat that it's making you hot sitting, 2 and 3 feet away from it. So the reason the low setting is there is because some incinerators will get plenty hot on the low setting. So when we're ready to sterilize the loop, it's the wire portion of the loop and not the aluminum handle that we're going to be putting into the incinerator. You can see that there is a metal plate on the front of the incinerator, and then there's a ceramic tube that runs down into the device. We're going to place the wire as far as it will go down into that tube for the purposes of sterilization. This picture is very blurry because I wanted to focus in on the loop as it sits way down in the back of the incinerator. You can see that the ceramic surface actually will glow red hot when the incinerator is ready to go. And the wire, at least the end of the wire where the loop is also glowing red hot. You need to achieve that in order to achieve sterilization. You've got to get that metal red hot. Now, as soon as you withdraw the inoculating loop from the incinerator, the red hot color of the metal is going to go away. It's going to go right back to its regular color. But understand that this wire is burning hot right now. So the very last thing you would want to do is take this loop at this moment and put it into your broth culture, because you will sizzle that broth culture and you'll kill a whole lot of bacterial cells. So instead, you just hold the loop in your hand and count to about 20. Just do a little mental count in your head to about 20. That's enough time for that loop to cool off to be safe to put into your culture solution. So remember, when it's time to take out the culture solution with our inoculating loop, we want to make sure that we're handling the culture tube correctly. Remember, always with a gloved hand. We don't handle any culture materials with our bare hands. And we're going to always remember, once we open a container that has pure culture on it or in it, we want to tilt it so we make sure not to allow any microbes to accidentally fall in. When you take the cap off, you want to keep the cap in your fingers. Don't put it down on the desktop. You're going to keep the tube at an angle while you draw out your specimen. You can see I've got the loop here. I'm not just sticking the loop right under the surface of the liquid. I'm going down about halfway into the tube. That's just good practice. It's also good practice not to allow the handle of the inoculating loop to get too far into that tube. And that's because, if you recall, we didn't sterilize this handle. We sterilized the wire, or at least the bottom half of the wire. So try not to put any more of that handle into your culture tube than absolutely necessary. Once you have culture on your inoculating loop, you can place that culture onto your glass slide. And you'll see I'm doing circle movements, getting ever wider, and basically spreading that liquid out across the center third of the microscope slide. Of course, we have to let the culture material dry. And all I've done here is I've set the slide down on a piece of paper towel. Notice that the slide is labeled. Typically, when you're making a slide that you plan on just looking at and then throwing away, the labeling is not important. But if you're making multiple slides, which is often what you're doing, you're going to want to label them. And certainly if you want to save the slide, if you're going to produce and stain a slide and you want to save it, you're going to want to label it. So I have my initials, I have the date that the slide was made, I have the name of the organism that's growing in my culture, and that is capital B for the genus Bacillus, and the species name is Subtilis. And then finally, I wrote MB on this slide because I'm going to be using Methylene Blue as my stain. The next step in preparing a smear for staining is critically important. That step is called heat fixation. We're doing a couple things when we heat fix a smear of bacteria or other microbes. Number one, we're going to warm up the surface of the slide so that the lipid in the membrane of those cells will stick to the surface of the slide. This is such an important step, and unfortunately, sometimes it gets overlooked. If you try to stain a bacterial smear that has not been heat fixed, when you go to stain that smear, you're going to wash the cells right off the slide. So we heat fix to adhere the cells to the slide. We also heat fix to kill the cells. Now, the staining procedure, the stain itself is going to kill the cells too. So it's kind of a double check that we've got the cells killed and stuck to our slide. Now, one other thing to know about heat fixing, and that's this: you never heat fix your slide until it's dry. So once you put your bacterial cells onto your slide and spread them out, you've got to allow that slide to dry completely before you heat fix it. And that's because if you lay a damp slide, a wet slide, against this hot incinerator like this, you run the risk that you're going to aerosolize that liquid very quickly, and you're going to send bacteria up into the air. And you don't want to do that. So make sure the slide is 100% dry, and then pick the slide up, preferably with a clothespin. And this is the clothespin I showed you at the beginning of the video this morning. The clothespin will allow me to hold this slide against the hot aluminum surface of my incinerator and keep my fingers far away. So you simply lay the glass slide right to the surface of that incinerator and leave it there for approximately 20 to 30 seconds. Once your slide is heat fixed, it's time to apply the stain. I'm applying a drop of Methylene Blue, again, to the center of the slide, because I know that's where I placed my cells. Now, don't be stingy with stain. You want to make sure that that whole smear that you took the time and trouble to make is covered with that stain. So put the stain onto the slide so that it makes a nice puddle. All right? Stain is not expensive. So go ahead and apply it generously. And that way you're ensured that all of the cells will get exposed to that stain. Typically, when we're doing a simple staining procedure like this one, where we're applying that one stain and then we're going to rinse it off, it will stay on the slide for about 60 seconds. Now, every stain is a little bit different, and sometimes it'll be a little bit longer or a little bit shorter, but generally it takes about a minute to stain a smear like this. Once that time is up, you get your stain waste container, like that bowl I showed you a minute ago. You're going to take that container, you're going to hold your slide over it, and you're just gently going to rinse the excess stain off of the slide. Now, notice you don't see a big blue area on my slide, even though it's been stained. You're not staining the slide, you're staining the cells on the slide, and you're not going to see where those cells are until you put that slide under the microscope. Once it's rinsed, you can allow the slide to dry. Just one more word about stain. All of the stains that we use in the microbiology laboratory have to be disposed of correctly. They have to be collected into a waste container and treated as hazardous waste. So the reason that we work in these containers, these stain bowls or staining trays, is because we want to be able to collect this waste stain and keep it from going down the drain. You can allow the slide to just air dry, but you can also use this material here, which is called Bilbus Paper. This paper is just it's a thick, absorbent piece of paper, and it comes in pads. So you use one of these pieces to just blot your damp slide and get it prepared for observation under the microscope. All I've done here is folded the Bilbus paper over, and I'm just going to gently press it down onto the surface of the slide so that I wick up as much of that moisture as possible. It's important to know that you don't have to get a microscope slide completely dry after you've stained it. If it's a little bit damp, that's not a problem. You don't want to rub the slide. You just want to blot it gently. You're trying to get up as much of the excess moisture as you can before you examine it. All right, very good. So that's just a very general procedure for preparing a smear and heat fixing the smear and then doing a simple stain with methylene blue. It was a nice little review for some of the things we've talked about so far, like cleaning off your work area and wearing your gloves and so on. There is one mistake in the video that I always have to remember to correct. When I'm talking about heat fixing, I don't know if you caught it, but I said that the heat warms up the lipid in the membrane. And what I just told you was that the heat actually warms up the protein in the membrane and makes it sticky. Okay, so just something for your notes. It's the proteins that are really affected by that heat. And remember, we said the enzymes, which are just a type of protein inside the cell, are also affected by the heat. Any questions about anything you saw in that short video? Any of the words you didn't quite catch or anything like that? That paper at the end, that's called Bilbus paper. I'm just going to write the word into chat so you can see it. Bilbus, B-I-L-B-O-U-S. It's really inexpensive. It's very thick paper. It's stiff, but it's very absorbent. So it's nice for drawing water off of a slide after you make it. All right, I'm going to bring us back in now to our slides. And now we'll take a few minutes and we'll look at some slides. We'll look at some stained slides. This slide was actually what was the result of the slide that I made in the video. So remember, this organism is called B. subtilis, and it was stained with methylene blue. And this is what it looked like when I put it under the microscope. Now, I am all the way up under the oil immersion lens here. So I'm under the 100x lens. And remember, we multiply the lens strength by 10, which is the magnification of the eye pieces. So these cells have been magnified 1,000 times. I think you can see that these cells are not round. They're not spherical. They're elongated cells. These are actually what we call rod-shaped cells. The scientific name for this shape is Bacillus, which is what the name of the organism is. It's called Bacillus subtilis. So
They kind of look like chromosomes before they come
yeah,
together.
yeah, a little bit, right? They have that look. They look like those stubby little condensed chromosomes that we sometimes see under the microscope. There's lots and lots of cells on here. You can see that there are areas where there are more cells sort of clumped together. And then there are some areas where we have single cells. But the important thing to notice is that those cells have been stained. They are now this lovely blue color. And the background didn't pick up any color at all. So that's what we call a direct stain. Methylene blue, remember, has a positive chromophore in it. So it's a basic stain. Here's another slide that was stained with another basic stain. The basic stain that was used on this slide is called crystal violet. Crystal violet is another very common stain. In fact, you're going to see it when we talk about gram staining in a couple weeks. Crystal violet also imparts a bluish color, but it's more purple, hence the name violet. It gives it this lovely dark blue purpley color. Now look at these cells. These are also rod-shaped cells, but they look different, don't they? There's this one, and there's this one. Some of what you're seeing here is kind of an artifact. It's not real. For example, it looks like one big long cell right here, but it's actually several cells that are all lined up, head to tail. So if you look right here, that's a single cell. If you look right here, that's a single cell. So when you see these longer formations, that's because these cells like to arrange themselves in chains. Remember, we said last time we were together that some types of bacteria like to sort of arrange themselves into different shapes. Here's another slide. Look at the chains of cells on this one. That's really impressive, isn't it? They line right up like little soldiers. And then they were attached to the slide and stained. Now, this particular organism, this is also under the 100x lens. So we're at 1,000 times magnification. I want you to look at this slide and then look at this slide. These cells and then these cells. Look how much bigger these cells are. They're enormous, relatively speaking, compared to something like this or something like this. Same magnification, just a giant looking bacterial cell. This is another kind of Bacillus bacteria.
Did those cells have a nucleus?
I'll talk about that in just a sec.
Okay.
You're seeing something in the center. This is a bacillus called Bacillus Megaterium, the mega part referring to the big size of the cell. And yes, there's something in the center of these cells. It looks kind of pink, right? Or at least it doesn't look purple. These cells were stained, and most of the cell picked up the stain, but something right in the middle of the cell didn't. And that something is a spore. Bacillus is a type of bacteria, and there are many different species of bacillus, but bacillus is a type of bacteria that can sporulate. It can turn into basically inanimate spores. There are two kinds of pathogens that are able to do this, that are able to sporulate and basically go into an inanimate form for a while. The genus Bacillus and the genus Clostridium. We talk about this in lecture, so don't worry about jotting it down now. But sporulation is a pretty neat trick if you can do it. It allows a cell that is experiencing stress in its environment. It allows it to go into a spore form and then just stay there until the environmental conditions get better. And the thing about bacterial spores that's really scary is you can't destroy them. You can't get rid of them with, you can't destroy them with disinfectants. You can't get rid of them with ultraviolet light. You can't get rid of them by just, doing normal everyday antibacterial things. You have to apply very strict and very rigorous tools in order to destroy spores. The other thing that's a little scary about spores is that they last, as far as we know, forever. We have found spores, for example, in the wrappings around Egyptian mummies. And if you take those spores and you put them back into a nice, warm and wet environment, for example, if you were to put them into some of this broth that we have in the laboratory, they will unsporulate, they will desporulate and go back to being living cells. So very interesting. And spores don't take up stain. And that's why you're seeing them without any color here. They will not take up the stain that the rest of the cell takes up. Now, you might ask, well, why are these cells stressed out? Because cells only sporulate when they're feeling stressed. This is an old culture. That's a stressful thing to a cell. In other words, these cells were placed into some broth in the laboratory, and then the broth was put in the incubator, and the cells were allowed to grow, to divide. But over the next couple of days in that test tube, they were using up all the nutrients. They were generating a lot of waste material, and they started to get stressed. In the laboratory, if we wanted to keep these cells going in culture, we would have to give them some fresh broth. Otherwise, they would sporulate and we wouldn't have living cells anymore. So if you haven't yet looked at the lecture material about the shapes of cells, don't worry too much about this, but this slide reviews for us these very basic shapes that bacteria take and that archaea take. There are spherical bacterial cells and archaeal cells, and we give that spherical shape a name. We call that a coccus. So when you hear the name Streptococcus or Staphylococcus as the name of a bacterium, you know that that bacterium has a coccus shape. It's shaped like a spear. I'm going to skip this one for just a minute and go to this one. This is that rod shape that I mentioned a minute ago. You can call these rods in this class because that's what most people refer to them as. But the scientific name is bacillus. We call this a bacillus shape. Now there is a shape, but it's unusual. there is a shape that's halfway between a coccus and a bacillus, halfway between a coccus and a rod. And that's what we would call a coccobacillus, sort of a oval shape. These are very unusual though, so don't worry too much about that. There's one type of bacteria that forms a rod that is shaped like a comma. And that organism is called Vibrio. So a comma-shaped rod. And then there are two bacteria that take on an elongated spiral shape. And again, I talk about this in lecture, but the longer one with more twists in it is called a spirochete. And the shorter one that has fewer twists in it is called a sporillum. That's what those two terms are. So the shape of this, in other words, is sporillum. The shape of this is spirochete. Now, most bacteria, just for size comparisons, most rod-shaped bacteria are 1 to 2 micrometers long. Remember, there are 1,000 micrometers in 1 millimeter. So 1 to 2. Now, some, for example, the Bacillus Megaterium can be 4 micrometers long or 5 micrometers long, but generally speaking, a couple of micrometers long. And again, we can see them under 40x, but we really want to go up to a 100x lens so that we can see them in the best detail. All right, let's look at some more. On this slide, you're looking at some bacterial cells under the high dry lens. Remember, that's the 40x lens. These cells were actually placed in a wet mount. So remember from last time, that means we just took a drop of the culture and put it on a slide and put a cover slip over it. But then what we did was we added just a drop of methylene blue at the edge of the cover slip, and we allowed it to be drawn under into the fluid. That's not the same as staining, where that's not the same as the simple staining process that we just looked at, is it? Because we didn't make a smear and we didn't heat fix it. We just added a drop of stain to a wet mount. Now, I think you'll agree that you can tell that there's some color to these cells for sure. You can see that they've picked up some of that stain. On this slide, all I've done is I've taken it up to 100x. I've taken it under the oil immersion lens. Now this slide is slightly out of focus because it was a wet mount. And remember, wet mounts don't have a nice single layer of cells. It's just a drop of the culture. So there's multiple layers of cells here, and they can be a little fuzzy to look at. But if you look, I think you'll agree that these cells are spherical. They're round. And they're kind of grouped together, some of them. Some of them are off by themselves, but most of them are in sort of clumps and collections. There's also a couple of things here, notice. There's something right here. There's something here. Does anybody remember what that is when you see that? Remember, we're under our 100x lens, so it's too big to be a bacterium.
Could that be RNA?
No, RNA would not be visible under a light microscope. I'm remembering something we looked at in the last class when we were talking about wet mounts. Remember the strawberry? Do you remember seeing something that looked like this? These are just little pieces of fungal hyphae.
Fungus? Yeah.
Yeah, it's just little pieces of fungus. So this was not a pure culture. This sample did not come from a pure culture. It's contaminated with fungus. But the little purple circles, those are bacteria. Now, why am I showing you this? This is what a wet mount looks like of coccus-shaped cells that have had a drop of stain added to them. And certainly I can see the cells. I can see them, But now look at what happens when I take that same culture and I make a real smear and put real simple stain on it. Now I get that. So wet mount with a drop of stain and a bacterial smear that is properly stained. Look at the difference in how much color gets taken out. You can see why people go through the process of making bacterial smears and staining them. You just get a better sample. You get a better sample. You can do a quick wet mount with a little drop of stain added in if you're just trying to figure out if bacteria are present or something. But you're going to want to make a nice smear if you're trying to do a real evaluation of the cells. So again, you can see a ton of cells on this slide. Most of them are all clumped up together. But in a place like this, you can see 1, 2, 3, 4 cells in a little square side by side. These cells are spherical. These are cocci. Coccus is the singular word and cocci is the plural. So they are a nice dark color because they've been stained with methylene blue. And if I needed, oh, sorry, go ahead.
I'm sorry, I was just wondering, would the fungus just be in the white background in this?
Oh, I actually, I cheated a little bit on here in that after I saw that the culture was contaminated, I actually made a fresh culture for the smear.
Okay.
So no fungus in here. If there were fungus in here, these would have picked up the methylene blue too. So you'd still see the fungal branches, the fungal hyphae in there, and they would be blue as well. Good question. Now here's another smear that was put through another simple staining procedure, but with a different stain, obviously. This particular stain is called Safranin. Safranin imparts this lovely sort of fuchsia pink color. I think you'll agree that compared to this smear, the cells on this smear really seem more clumped up together. That's not an artifact. That's actually a feature of this particular organism. If you look at a place like right here or right here where you see two cells side by side, you can see that this is a round cell. This is a coccus, but it's definitely more clumped than these cells were. And that's a feature of this particular organism. This happens to be Staphylococcus. And Staphylococcus likes to do this right here on a slide. It likes to really just clump up together. But look at that beautiful color. I know I'm geeky. I know I'm a nerd, but look at that gorgeous Safranin color. Just a different stain, a different color, but it's doing the same thing. It is imparting one particular color to all of the cells on that slide. Here's another one. This was also stained with Safranin. Obviously, these cells look very different. We're under the oil immersion lens again here. So we're at a total of 1000x magnification. These are rod-shaped cells, and these cells like to form chains. These are very, very small rods. Let's go back for just a second. Remember, that's under 1000x magnification. And this is under 1000x. These rods are tiny, very, very narrow, very thin. That's the thing about bacteria. Each genus and species of bacteria is going to have a different size, even if it has the same shape as other species. So just because a bacterium is a rod doesn't mean we know exactly how big that rod is going to be until we look at it under the microscope. Here's an interesting looking slide. We're under the oil immersion lens here, so we're at 1000x magnification. Look at these cells. These cells are wavy. They're elongated. They look like really long rods. These cells are not in a chain of cells. This is one cell. This is one cell. These are spirochetes. Spirochetes are long, and they have these waves in them. This is a particular kind of bacteria called Borrelia Burgdorferi. How you like to spell that name, huh? Borrelia Burgdorferi is the bacterium that causes Lyme disease. So this is a pathogen. This is a bacterium that causes us a lot of trouble. Now, this is also stained with safranin, but I want you to notice that unlike this slide, on this slide, the background has turned pink. Do you see that? Remember, direct stains like safranin should only stain the cells and not the slide, but we've got the slide picking up stain here. And that's because this sample is not just from a culture. This sample was taken from somebody's joint. This is actually joint fluid. So they stuck a needle. I hope I'm not upsetting you by saying this, but they stuck a needle into someone's joint, perhaps a knee or an elbow, and they pulled out some of the synovial fluid and they put that on a slide. And look, there's bacteria in there. Lyme disease can be a terrible disease if it's left untreated. So that's why you've got staining in the background, because synovial fluid has protein in it and things like that, and it's picking up some of the stain. This interesting looking image is a spirochete that has been examined with an electron microscope. The reason I put this in here, number one, you can see how huge it is under the electron microscope, but I want you to notice how many twists it has. Spirochetes have lots and lots and lots of twists, number one, and they're very flexible. So when this cell is just living inside the human body, it's very flexible, like a piece of spaghetti. That's different from a sporulum type of bacteria. And that's what you're looking at on this slide.
Could slide 19 be like similar to like a tapeworm?
You have to remember sizes. So a tapeworm is going to be grossly visible, right? A tapeworm, you're going to be able to see with your eyes, as disgusting as that sounds. And this thing is microscopic. This is a single cell, even though it looks really long. This is one cell that just happens to have that long, long length and all these twists in it. Good question. On this slide, these are the shape that we call sporilla. Sporillum is how you say one, and sporilla is the plural form of this word. So notice this shape is also curved, but there's only one or maybe two twists in each cell, unlike these, which have many, many twists per cell. The other thing about bacteria that have a sporilla shape, they're not as long as spirochetes, and they're much stiffer. They don't have the same sort of spaghetti softness that spirochetes have. So they're very similar in their shape, and they're easy to confuse. But a spirochete and a sporillum are two different shapes. Any questions? There are other spirochetes that are not pathogens. There are other sporilla that are not pathogens. But that Borrelia Burgdorferi, is a very bad pathogen. In fact, I just saw an article today suggesting, in a journal that I was looking at, suggesting that Lyme disease, particularly in the Northeast, is getting worse. That we're having more and more and more cases of Lyme disease every year. And the scientists who did the study suggested that it's probably a reflection of how the climate is changing, because we're not getting as cold winter as we used to get. We're not getting a long period of very cold temperatures in New England anymore. I can remember, for example, when I was little, I can recall winters where, all the way through January, it didn't get above 0 degrees. The problem now is that we have some winters that are quite mild. We may never go below 0 through the whole winter. And as a result, you don't get a good die-off of all the ticks. It's hard to imagine that there are living ticks outside right now, but there are. They go down underneath, pine needles and old leaves, and they get some cover from the weather, and they survive the winter. And so scientists are very concerned that that particular pathogen is getting worse. It's becoming more and more prevalent because of perhaps the changes we're seeing in the climate. Very scary. The only good news about Lyme disease, as far as I'm concerned, is that it is treatable if you catch it early. We can treat it, we can get rid of it if you catch it early. All right. Just a couple other things to go through with you here. Take a look at this slide. This is a clinical specimen. This is urine sediment. So this came from a patient. We collected a urine sample from this patient. The urine was spun down with a centrifuge to create a sediment. And then the sediment was placed on the slide. And this is the result here. Now, this is a wet mount. This has not been stained, but we are at 1000x. And under the oil immersion lens, what do you see? Do you see cells, number one? And what shape are they?
Caucus.
Caucus, very good. Does everybody see them? They're hard to see because they're not stained. But here they are, these little circular cells. So there's a caucus in this urine. This person has an infection. Here's another one, also urine, also a wet mount, no stain. Do you see cells? And if you do, what shape do you think they are?
Sporulum.
That's a good guess. If I looked at that right there, I would say sporulum.
Rods.
These are actually rods. Now, I only know that because I have, the ability of knowing what the rest of the slide looked like and you don't. If you called this sporulum, I would absolutely give you credit for that. They look like sporulum, but it turned out these were rods. One of the things I put this slide in for, because I want to remind us, is that when we look at a slide, we're not going to always see the cells from the side. So we're not going to always see them along their length. You have to use your imagination here for a minute, but sometimes you're going to see them end on. So for example, see that circle right there? You would think, oh, well, that's a circular cell. That might be a caucus. Maybe there's two kinds of bacteria here. What you're looking at here is a rod-shaped cell, but we're looking at it end on. So in other words, think about a loaf of bread. You can look at a loaf of bread from the side and you can see all the slices. Or if you rotated it, you could look at the loaf of bread end on and you would only see one slice. That's what happens a lot of times when we examine these microbes. So you have to look around. You have to look at all the cells to get a feeling for what the shape is. If this slide was truly a caucus-shaped bacterium because of this, I wouldn't see something like this. I wouldn't see something like this. I wouldn't see something like this. So it has to be either a rod or a sporulum. Or, like I said a minute ago, it is also possible, since this is a clinical specimen, that there are two shapes of bacteria on here, or maybe even three. It's not uncommon to have mixed bacterial populations in clinical specimens. Here's another one, just an interesting case. This is also urine sediment. This is a wet mount. We're under the oil immersion lens, no stain. What do you think this is? Have you seen this before? Did you see this the other day with me?
Is it yeast?
Yeah, this is a fungus.
Yeast.
And it's budding. The yeast cells are budding off of the hyphae. This is a yeast. Interestingly enough, this patient, this was a routine urine specimen, and this patient did not have any symptoms or signs of a UTI, a urinary tract infection, and was not diagnosed with a UTI. So let me ask you, how is that possible? This is in her urine sample. So if it wasn't in her bladder, where did it come from? What do you think? Any idea?
Could it have been contaminated when she took the sample?
Yeah, very, very common thing to happen. When we collect urine from a human, remember my veterinary background, we ask people to urinate in a cup, right? You may or may not have done this sort of a collection before for yourself. And we do ask people to clean off their external genitalia with some alcohol wipes. But when you catch urine passing out of the body, it's very common for the urine to contain some microbes that are not from the bladder, but they're from the skin around the outside area, from the perineal area. So it's very common to see an occasional strange thing in a urine sample. And when people evaluate urine in the laboratory, there's a little bit of art in it, in addition to a lot of science. You have to know whether or not that person honestly has a yeast infection in their bladder, or whether that's just a contaminant from the sample collection. And one of the ways that physicians make that decision is, well, based on symptoms, number one. This person didn't have any symptoms. And number two, the amount of microbe. The chances are, if that woman had a true fungal infection, a yeast infection in her bladder, the slide would be covered with yeast. And that was the only yeast that was found on the sample. So she didn't have a yeast infection. She didn't have any problems in her bladder. We just saw the yeast because it was rinsed off of her outer skin as she urinated into the cup. Remember, we are covered in microbes, including fungus. Very, very common for us to have fungus on our skin, out on the outer part of our genitalia and in our perineal region. Very, very common. And it's not a problem. It's not a pathogen. It lives there. We sometimes don't like to believe that we're covered in microbes, but we really are. And they don't cause us any trouble at all where they normally live. I'll also mention here, because it is an important thing to know about microbiology in terms of medicine. I mentioned in the lecture that the two microbes that cause most disease in humans are bacteria and viruses. The fungus just aren't as big of a problem with humans. Now, that doesn't mean humans can't get fungal infections. They can. But it just turns out that fungal infections are less common than bacterial infections and viral infections. The people who tend to get really bad fungal infections have something else wrong. Think about a burn patient. If you've been horribly burned and you've lost a bunch of your skin in an area, you've lost a barrier. And those people are very susceptible to yeast infections in their skin. People who have underlying health issues, like patients who have HIV, often get yeast infections in their mouth. And again, that's because their immune system is under attack by the HIV virus. And so they can't fight off fungus as well as healthy people can. So it's a good thing to keep in the back of your mind that most pathogenic disease-causing microbes in humans are bacteria and viruses. All right, just one more thing to finish up with, and then I'll let you go. This is a clinical specimen. This is a Buffy coat smear. This is white blood cells and something else. These right here are white blood cells. I want you to notice how big these are compared to what's sitting next to them, which is this. This is bacteria. Look at these big cells. These are eukaryotic cells. These are human cells. And relatively speaking, they are much bigger than bacteria. Now, this happens to be that horrible bacteria that we looked at earlier called Bacillus Megaterium. So these cells, these bacterial cells are big compared to most bacteria. But even at that big size, they're much smaller than these white blood cells. Do you think this patient is sick? Do you think this
Yes.
patient
That's a
has-
lot of white blood cells.
Well, it's a lot of white blood cells, but it's also
A lot
bacteria in the blood.
of
right?
bacillus. Oh,
Yeah.
okay.
If
So
you're not,
can I ask another question?
yeah,
Can I
yeah.
ask a question? Is bacillus have like, what does the root mean of that? Is that like
Oh,
skin
so-
or like the base?
So the word bacillus means rod-shaped or rod. So these bacteria got their genus name. Remember, all bacteria, well, all living organisms have two scientific names. They have a genus, that's the first name, and they have a species, that's the second name. So you and I are Homo sapiens. So we belong to the genus Homo and the species Sapiens. Every living organism has two scientific names. So those bacteria got the name Bacillus because somebody looked under a microscope and saw that they had a rod shape. And they said, oh, well, Bacillus. The Megaterium name came from the fact that they're so big. Now, anytime we find bacteria growing in the blood, that's referred to as sepsis. That's a bad thing. That's a bad state. That person is not only sick, that person's very sick. If you're not familiar, I'll just briefly tell you that slide that we just looked at, that came from what's called a Buffy coat smear. What we do is we draw blood from the patient and we spin the blood in the collection tube. So all of the red blood cells go to the bottom of the tube. The white blood cells go to the top. And when you look at a blood cell tube that's been allowed to clot and has been spun, the layer on the top is white and the layer on the bottom is red. And the reason it's white is because it's full of white blood cells. So what we've done is we've collected all of the white blood cells in what we call the Buffy coat. I don't know where that terminology comes from, but a Buffy coat is the layer of white blood cells that you get in a blood collection tube after you spin it. So they took a little bit of that material, that Buffy coat, and they smeared it on a slide. And that's why you see so many white blood cells on that slide, because we've purposefully concentrated all of the white blood cells there. And you can also find the bacteria in the Buffy coat. So that patient would have been very sick. That patient would have been in sepsis. Just a couple of clinical things. I try to add some clinical things in for us so we can see microbiology in action. Anybody have any other questions? Any questions about simple staining? Any questions
I have
about?
a technical question.
Yeah.
Can you hear me?
Yep.
Oh, hi. Okay, so if you know that you suspect things are going to move and you make a wet mount, procedurally, if you know you're going to have do a simple stain as well, when you're getting your specimen out, can you drop the the broth from the loop onto 2 slides at once?
Oh, absolutely.
If
And in
yeah,
fact, if you
So
know
you're
if you know you're gonna do a simple stain, you'd probably skip the wet mount unless you needed to know if it was mobile.
yeah, that
You
was my
do your
question.
wet
If you know
mount.
you're gonna do 2, just start 2 at the beginning.
Yeah, and that's the best way
on.
to do it. So when
Cool.
you watch a technician in a laboratory who works in a micro lab and they get a culture tube in or a patient sample in, they're going to lay out all the slides they need to make. And they're going to put cells on each one and they're going to heat fix all of them. And then they'll do the different stains that need to be done, or they'll do the wet mount. Yeah, absolutely do several slides at the same time because it just makes it easier. It just makes it more efficient. Yeah. And remember, even though you have to do a wet mount or a hanging drop if you want to find out if the bacteria can move, we also just use wet mounts just to find bacteria sometimes. Sometimes we just do a quick wet mount. We take a drop of the urine sediment, we put it on a slide, we put a cover slip on, we look under the microscope. Sometimes we're just interested, are there bacteria in there? And before we go to the process of making a smear, we just do a quick wet mount and look. Yeah. So you can use a wet mount even if you're not interested in motility. You just want to see if there are any bacteria in a sample. But then you're going to follow that with a smear. Very good questions. Oh, and the other thing to remember is, remember we said one of the drawbacks to a wet mount is that it's not going to last because it's going to evaporate. Once you make a smear and you stain it, you now have that slide. That slide's going to last. That's one of the reasons it's so important to label it. So you can keep that slide. You can give that slide back to the physician if the physician wants to take a look. You can give it, to other people to look, but you can save it. Remember, the cells are dead at that point. There's no danger to anyone at that point. The cells are stuck to the slide, so they're not going to fall off. And we can keep stained slides, which is why I had the slides to show you today in the presentation. So that's another reason why it's really nice to make a smear and stain it. All right, very good. Any other questions? All right, I'm going to set you free, put you back to work. Remember, you have a quiz due tonight. No, a quiz was due last night. And you have or 2 quizzes were due last night. Let me see if I can get back on schedule here. And then you have another quiz that's due Thursday night. And don't forget, you have your lab questions to do for Sunday. Okay. You guys are doing great so far. Keep up the good work. And I will see you again on Monday. Don't hesitate to email me if you need anything. Okay. All right. Take care. We'll see you next time.
Thank you.